Hi and hello everyone. I am Partha from Biomix team. Welcome you all for today's session. Education breeds confidence. Confidence breeds hope. Hope breeds peace. Education is either pulling of learning, information, data, facts, skills, or abilities, not just about going to school and getting a degree. It's all about widening your knowledge and observing the truth about life and making visible what is hidden as a seed, says Confucius. Today's webinar on research methodology and application of surface plasmon resonance is brought to you by the Biomix team. The speaker of today's webinar is an eminent resource person, Dr. Prashant Pradhan, PhD, who is an assistant professor in the Department of Biomedical Sciences from the University of Delhi, India. He will be imparting advanced knowledge on the research methodology and application of surface plasmon resonance in biology. At this juncture, I would like to thank Professor Dr. Prashant Pradhan for taking the time out of his busy schedule to join with us through this webinar. It's a highly honor and pride and privilege for our Biomix team in hosting this webinar with an eminent resource person. This webinar enriches you with the current knowledge of major points of one to think on before joining any research laboratory, to know the fullest potential of yours as well as your neighboring research institutes, and he will help you in selecting PhD topic for your research and adding new techniques to your curriculum vitae. And Dr. Prashant Pradhan will be elaborating the applications of surface plasmon resonance in biology as well. We must welcome the future, remembering that soon it will be the past, and we must respect the past, remembering that it was once all that was humanly possible, says George Satania. I am very glad that Biomix steps up to new heights in organizing a series of webinar talks. On envisioning our motto, this webinar talks are the new initiatives on our Biomix platform. Biomix has successfully completed 2 webinar talks so far and steps up in his 3rd webinar talk today. On behalf of Biomix team, I would like to welcome our eminent resource person, Dr. Prashant Pradhan, PhD, who is an assistant professor in the Department of Biomedical Sciences from the University of Delhi, India. Our speaker is an eminent scholar in the area of biomedical sciences. Henceforth, I believe that this webinar would be nourishing the young minds in the field of surface plasmon resonance in biology. Now I extend my warm welcome to Mr. Sachin, research scholar, Indian Institute of Science, Bangalore, and Vishal Kumar, who are the admins of Biomix. And I also appreciate and welcome all the scientists, deans, professors, principals, and students from various colleges, universities, and research institutes globally who joined with us through this webinar. Our eminent resource person, Dr. Prashant Pradhan, is an assistant professor in the Department of Biomedical Sciences from the University of Delhi, India. He has completed his PhD in neurobiology from Indian Institute of Technology, IIT Delhi, and he did his master's in MSc Biotechnology from South Asian University, New Delhi. He is currently contributing to the medical sciences with the help of surface plasmon resonance through his research. Dr. Prashant Pradhan is an inventor of world's most affordable RT-PCR based COVID-19 diagnostic kit, which has been e-launched by Union Human Resource Development Minister Sri Ramesh Pokhriyal since this pandemic. COVID-19 diagnostic kit developed by Dr. Prashant Pradhan is a step towards Prime Minister Sri Narendra Modi's vision of self-reliant India. This kit is a cheap and reliant testing for the countries which can help to control COVID-19 pandemic. The COVID-19 pandemic kit has been developed indigenously and is much cheaper than other kits, which ensures a healthier India, especially in the times of COVID-19 pandemic, with this innovative research. The kit has received ICMR approval with the highest score and DCGI approval with a very high sensitivity and specificity of 10%. This kit, world's most affordable RT-PCR based COVID-19 diagnostic kit, which has invented by our eminent resource person, Dr. Prashant Pradhan, has significantly brought down the cost of COVID-19 RT-PCR testing. Even after adding the RNA isolation and laboratory charges, the cost per test will be considerably cheaper compared to other available kits in the market. IIT Delhi has given license to 10 companies to manufacture COVID-19 diagnostic kit using this technology, which has been developed by our eminent resource person, Dr. Prashant Pradhan. SAR has also recently contributed towards the research on coronavirus in this COVID-19 pandemic through its research article titled Unscanning Similarity of Unique Inserts in the 2019 Coronavirus Spike Protein to HIV-1GP120. He also his recent research on the prion protein for the treatment of fatal transmissible spongiform encephalopathies, which is also highly recognizable and recommendable. Dr. Prashant Pradhan has qualified CSIR NET as well as GATE examinations. His area of interest in research falls in prion biology and nucleic acid, protein and RNA interactions and secondary structures of DNA. Now I would request our research person, Dr. Prashant Pradhan, to have more insights on research methodology and applications of surface plasmon resonance in biology so that the enhanced understanding could help us better save our country and its people through research and development in the field of molecular biology. Before that, I request the audience, if you
I'm audible.
yes, you are audible, sir. You can proceed.
So thank you, Partha, for such a good intro. I myself realized now only that I have done this by your introduction, and it's good. And I would like to thank all Biomic team member to provide me to give an opportunity to speak or to talk with young minds. And thank you, Biomics, for this. I'm sorry that something went wrong with my laptop. I'm using my phone, so might be I will not be able to use cursor to explain some of things, but I will try my level best to make your things clear. So, Partha, my screen is visible.
Yes, is it visible? You can proceed, sir.
Okay. So, when Partha told me to give some talk related on COVID-19 or other this kit, so at this time I thought that instead of giving talk on this, I should speak on a topic which is very important to every candidate who wish to do PhD. So, and generally we have a lot of questions when we are in MSc or in last year of our BSc, that what type of lab we want and what institution will be good. So I will be majorly talking about this, and then I will be talking about SPR, surface plasmon resonance, because I did some experiments, or mostly my PhD was based on SPR. So I will be talking about the applications of SPR and the other techniques that will be helpful for your career. So to start with, so what are the major points to think before joining lab? So there are, the first thing that when student gets an admission in any institute, he generally talk to their peer group that should I join that institute or not? So some students tell you like, oh, you got admission in IIT. Oh, you must go. IIT is a big place. You will, it will help you to make your career, or IISc, Bangalore, or any IGIB, or CCMB, CTFD, any big institution. So, is it true that we should always consider the name of institution, or there are some other factors? So to start with, when you get a lab, you must always first look that the weather. So I guess the first point is visible in bottom of your left screen. But the funding in lab, the first point you must always consider before you are joining a PhD in any lab is that how is the funding is present in that lab, because Many times it happens that PI who is taking you probably has written a grant and is waiting for that grant, and he assumes that that grant, and he is very hopeful that that grant may come, and on that basis he is going to take you, and it is good. But if you don't have any running project in his lab, then you have to be critical about it, because funding is the important thing that you must always consider before joining lab. That what are the running projects, what are the projects which has been written to get the funds if it is not present in lab, and what are the chances of getting it. So because many a time student gets into a lab in good institution, and they struggle for funds, and without funds we know that research is not possible. Then second point, projects going in lab. So what we do is that we think that, okay, I got a lab, I got a lab which is very renowned, and because of this, the student believes that I must join that lab. But the criteria should be that what are the projects which are going in lab. So does that project gives you kick gives you kick so that you are willing to join that lab. So what all type of projects are going is that the lab is focusing on a particular area, or a lab works in different domains, such as the lab has funding on so lab works on bioinformatics, lab works on drug discovery, lab works on biophysics, lab works on cell culture, and then lab works on a lot of high throughput techniques. So what all are the projects, and what all things or techniques that lab is utilizing to accomplish the goal they have laid down. So because this is important, this becomes very important, because a PhD is just a learning process, and the learning is incomplete when, especially in biology, when you are not able to link or get enough exposure to use techniques which will which help you to make your paper more presentable and more in a way in a more so let's suppose generally reviewers when you write a manuscript, it goes for review, and many times reviewers says that, okay, you have shown this result from this technique, but it will be good if you can show the result with other technique. So it's always good to be in a lab, or where multiple projects are going, so that multiple experts will be there, your seniors will be there, which can help you, who can help you to get rid of those questions which has been raised by reviewer. The third point is personal attributes of PI. Now, first point was very important. Second point is, suppose you got a lab which is well funded, projects are very good. Then what about the personal attributes of PI? This also becomes very important, because many a times, so you have to talk, you must talk to your prospective or the seniors who are present in that lab before joining lab, or any student in the department, that whether that PI gives time to the lab, whether the PI is collaborative in nature, whether PI is not very insecure. Because if you see in research generally, now the trend is changing, that like people, PIs were initially were not too much collaborative. Nowadays, the scientists have realized that if you want to do a 360-degree research, it's very important to be collaborative. But still, there are few people who believe that getting master in one technique and doing or improving things through one technique which is present in lab is well enough. So in that case, the PI will not be that much collaborative. And if PI is big and if PI is very renowned, in that case, he may be busy in a lot of meetings, and it is good to be in a renowned lab and with big PI. But it is equally important to know that whether the PI is too busy, whether he or she gives enough time or sufficient time in lab to discuss the projects, to discuss problems that are arising, and to give inputs. So this is very important. So the first 3 points are very important: to get a lab with good funding, uh, to know the projects, what are the different projects that are going in lab, to get a good project with your PhD, and then what are the personal attributes of your PI. Now, if let's suppose you were lucky, you got all three things: good funding, good PI, and good project. Then it's always good to connect with students which are currently and previously worked in lab, because they are going to help you for another 5 years, because they will be there, or at least for another 3 years or 2 years. And believe me, making a harmonious and good relation with your current lab member is very important to excel in PhD, so that, because they are the one who are going to help you, to teach you, to guide you in every step, because you will interact with your PI once in a day or twice in a day, but you will interact with your lab members daily, and they supposed to be the true immediate guide on which you can always talk to them and can get useful information. And then, what are the facilities in lab and department? So before joining, so let's suppose you get 3 options to join an institution for your PhD. So you must look which institute has good facilities. So that when I say facility, I mean what infrastructure in terms of equipments they have. So suppose you are going to join you are interested in a protein engineering or a protein biology lab. So it becomes equally important to see that whether the lab is fully equipped with techniques such as the circular dichorism. Let's suppose if you want to use that technique like CD, flow spectroscopy, NMR, for biophysical binding characterizations like isothermal calorimetry, ITC, SPR, surface plasmon resonance. So what all facilities they have? They have good microscopy facility. They have good in a way good cell culture facility. So please check all these facilities if you have good options to join any institute. So if you have 3-4 options, then you must consider this as a very important key point, that if you have number of facilities or equipments in your lab or in your department, it will be always great to add those or to learn those facilities to your CV. Then collaborations of institute. So better in nowadays, better the institute is connected, better it is for your growth. It's because is because having collaboration makes you to build a network in our field, and which nowadays is very important, because in biology, in any field nowadays, it is just like having a good collaboration will give you more exposure to things, and they also helps you to access the facilities which are present in their institution, and it also helps you to get good reviewers for your manuscript, because the students who are in UG and PG, they might not be aware of this thing. When you write a research article, and you send it to to get your manuscript reviewed, you have to give at least three reviewers name or five reviewers name. So when you have a good collaborations, when you have good network, then it will be easy. So you can give their name as a potential reviewer, and God knows if that person becomes a potential reviewer, it will be good for you. So it is also important to have to know that is whether the faculty or the PI you are going to join is not paranoid. I mean, the person is not interactive with their own faculty members, and in that case, you will be in somehow in a lost condition, because in losing condition, because if your PI don't have a harmonious relations with in his or her department, then you may not be getting the facilities that other department or other lab is having. So it is also equally important to know everything in and out of because it and why not? Because when PI interviews you, they want to know everything about you, that what you have done, what is your so they ask recommendations from your previous teachers, the scientists who have taught you. So it becomes equally important to know about the PI and to get the recommendation of the PI from other students or faculty members. And it is not wrong. And some people take it as wrong, but it is not wrong to know everything about your lab, your seniors, what is the funding, what are the projects which are going. You must know everything before joining a lab, because you are going to invest another 5 years of your life in that lab. And believe me, if things are not good, if the things are not harmonious, then it is sometimes it becomes very frustrating, and people think of quitting lab in some extreme conditions. So this is the same point about faculty of his or her willingness to work collaboration, because if the PI is very collaborative, it will be very good for you to learn, to go even in the places in India or abroad. And I have been to Taiwan and USA to do some part of my research, because my PI was very collaborative and he was always open to talk to people. And it's not only the PI has to be collaborative; it is something that you people also have to develop. That you people should talk to more and more scientists in India or in abroad, because the scientific community is very welcoming in terms when you talk to them, when you ask problem, when you discuss any kind of result or any technique that you are not able to understand. In many cases, I say, like in 90 to 95%, in my case, I was fortunate enough to get a reply, and they were very helping to make me understand what is my result, how I can interpret my result. And so it becomes very important for you also to develop this character of collaborating and discussing to students, because sometimes people are so much insecure, students are insecure that they do not talk about their research, so that they may steal your idea. But believe me, people have a lot of things to do, they have a lot of other stuff to do, and it's always you have to talk your clever manner that okay, I am facing this problem in this technique. You must not tell everything about your project, but you can tell that in what way you are stuck. So I'll skip this point. Now, this scholarship funds to attend conferences in India or abroad. So if let's suppose again you get pre-institute, a good university, a good institute like IIT or IASC. So there are institutions which gives internal funds which support their student to visit abroad to attend conference, workshop, or to do some training. So, like in case of IIT or ISC, I also believe in ISC also, that they in any MHRD institute, they give 1.5 lakh in IIT Delhi, they give 1.5 lakh to attend conferences or workshop or to visit the lab you want. And Additional 1.5 lakh is also given to students who exceptionally do good in their PhD and committee recommend. So in short, they get 3 lakh lump sum rupee to visit multiple places. Apart from this, I want to tell students who are planning to do PhD that you during your PhD you get a contingency of like somewhere it is 20, somewhere it is 30 or 50,000 to 50,000. You must utilize this contingency to attend workshops, seminars, or conferences which occurs in India, because there are a lot of conferences which goes in India where you can visit those places, you can talk to people, you can build your network, you can understand what are the new techniques, what those people are using, and try to fit your thing into that in those facilities if you can. It's not always necessary to fit your project in that line, but if you get that chance, you must do, because that is going to increase the CV or the techniques that you have learned. And believe me, that getting a post or getting a faculty position nowadays, it depends on that what new technique or what new thing you are going to take with your with you to that institute. So what add-on you are going to give to the institute. Any regular thing which every faculty member is doing, believe me, most of the time committee do not prefer, because they want something new, a new add-on to the existing technology or the knowledge they have. So, Partha, I guess this point of first is like the first point of my talk is done. So, should we take the question later on or now also? Hello. Hello.
Yes, sir. Yes, you are audible, sir.
Yeah. So should we take the questions for this right now or later?
Later on. Not
Okay.
now.
So because I got scared that I'm not audible to all, and I'm
No,
speaking
you
to
are
myself.
audible. Proceed. You can proceed, sir.
Okay.
Sir, your voice is breaking. So that I think so there is a problem with your bandwidth of network.
So I guess I'm connected to my Wi-Fi. Okay. So is it audible now?
Is it audible? When you are lecturing, your voice is getting broken.
So it was frequent,
No,
or it is in like sometime it has.
You can proceed. Yes.
Okay. So potential of neighboring institute. So when you join any institute, let's suppose you plan to join any institute, and now you are there. You got a lab which is well funded, very good PI, very good lab environment, and everything that was there in first slide. You got everything. Now it's your duty. It becomes your responsibility to know the potential of your neighboring institute. Let's suppose I was in IIT, and IIT is surrounded by ICGV, NII, JNU, Jamia, Delhi University South Campus, ACU, South Asian University. So I was aware of the faculties that are used in my field, and those who are having techniques which are not present, or the facilities which is not present in our department. So it becomes very important to know the faculty members, to know facilities which are there in neighboring institute, because many times we feel that, oh, where I will do this this any method that is asked by your reviewer, or any research project comes to your mind. So in this case, it becomes very important that if you have any faculty in your neighboring institute, you can go there easily instead of going to any institute in India, which because in commuting you may lose 10 to 12 hours, or even your sample is not that much stable, so that you can travel along with that sample. So it becomes very important that you must know the facilities and the security members and the techniques that they are using and the facilities they are having, so that you can approach them and you can just do your experiment. So you plan to make a symbiotic relation with these institute, and this will always going to help you, because you are not the only one who is going to get help from them. Sometimes, or in fact many times, they will also ask you that, oh, like you are coming from that place, you have this technique in your department, you know that, so can we use that? So in this way you will get an opportunity to collaborate with that lab in a symbiotic way, and you can and many a time people get authorship in their manuscript also. So in this way you will get opportunity to get number of good manuscript where you are also contributing in their project, and this helps you to build your CV, to build your hello, yeah, so to build your CV. Now I'll talk about PhD topic. So when my juniors, when I became senior in my lab, so juniors usually come in lab and they ask that, what PhD topic should I take? Okay, and because they are naive and they do not know that what will be for them. So that's what I told you, that it's always important to build a good relation with your current lab members and with your seniors, because they are going to tell you that this project is running very good in lab, and they have a potential to get more paper, so that you can get more papers or paper early in your PhD, so that you can finish your PhD as soon as possible. So in this regard, wisely choose a project if there are many. So choose a project where you are going to learn a lot of things, instead a project which is very limited, like a project only involves a cell culture or a Western blot, or a project involves a cell culture, biophysics, bioinformatics, or any high throughput technique. So try to get yourself involved in a project which is using number of technique, because I will repeat it again, that PhD is just a learning process. Now, what is the scope of project, feasibility, and the facility to accomplish it? So the scope of project is that if you get a project, then sometimes the project looks very lucrative, and that you have every facility to accomplish that project, but the project should be feasible enough that the project should get completed in your 5 years. Sometimes people take projects which are sometimes people take a project which takes a lot of time, and eventually they get frustrated that within 5 years they were not able to complete it. So make the project or the objectives that you are going to the objectives you are going to choose to do some part of your project very wisely, that it must get completed in your 5 years. Again, I will say that PhD is just a learning process. Sometimes people spend, I have seen that they have spent around 10 to 12 years in their PhD, and later on they face a lot of problems in getting a good postdoc, or not postdoc, getting a good faculty position, because they have spent a lot of time. And if they get a good paper out after even 10 years, then again you have lost a lot of time to get that paper. Instead, if you have waited for a postdoc after 5 years, you may get a good papers during that time, and you also now more experienced with your postdoc exposure. Now, have 360-degree approach for any project. Try to add any high throughput technique to your CV. It is very important that in your PhD, you must you must learn a technique which is not many people know in your department, so that you become an expert of that technique. And if any collaborator, potential collaborator, or if any person talks of, oh, if anybody knows this technique better than us, then they must know your name instead of others. So you must learn a technique to the core. Whatever you are learning, learn it to the core, and number of techniques. That's what I was telling you. Attend workshops. There are a lot of good workshops which are done by which are organized by NCBS and IASC in terms of training. You must attend those training programs, even if you have to pay 20,000. Then I guess they are worth it. And generally, you get contingency, so you can use that contingency to utilize to get exposure to these workshops. So when I say 360-degree approach, I mean, so let's take an example here. So let's suppose you are interested to know about a gene. So a gene is a single thing. It's just a gene X. So students feel that, oh, what I will do in this? What is what all I can do to know about gene X? First is bioinformatics. Nowadays, bioinformatics is very important, becoming very important. So let's suppose you got the gene X. Now, to get a paper early in your PhD, bioinformatics is very helpful, so that you can do some bioinformatics. You can use some bioinformatic tool to find some lead molecules, and you can build a good phylogenetic tree to know that what is the evolutionary tree of the pattern of this gene, what are the mutations which are known for this gene, and you can collect all these information. Even you can write a review. So bioinformatics becomes very important nowadays. So you must learn this some bioinformatic tool, because there was lockdown and most of you are in your home for next other 2 months also, because institutes are not going to open at least for another 1 month. So you must learn some coding and some bioinformatic tools. So because they are the one which will going to give you edge and give you early paper. Now, when I talk about 360-degree approach, I will open everything. And then you will realize that I was trying to tell you. So first on your left side is transcriptomics. So let's suppose you did some good bioinformatics. Now you can always look that what are those miRNA which can regulate my gene, or that what happens to the RNA if gene express it forms RNA. What is the stability of that RNA? You can do some experiments on that, and you can do some RNA sequencing to know that if my gene get overexpressed. So let's suppose I overexpress gene X or I delete gene X, what is the change in overall RNA level? So people do microarray, people do RNA sequencing to know that what are the changes which are there if I delete gene X. So I'm just telling you that what are the number of things you can add to gene X. If suppose you get a project on gene X. Hello.
Yeah, you can proceed, sir.
So there was some voice.
Okay. No.
Is there any question?
Sir?
Is there any question?
No, sir. Sir, let me know before you are starting a surface plasmon resonance. We can complete the questions and then we can jump onto this.
It's just the last slide of this topic.
Okay,
And then
yeah.
then you can look into the proteomics part, that what all are the interacting partners of the protein which my gene X makes, what is the function of this protein in presence or absence of stress, what happens to the levels of this protein in the presence or absence of stress, what are the post PTMs, what are the post translational modifications that happens to my protein. So PTM, identifying PTMs, you can get you a good paper, and get knowing the interacting partners of your protein gives you a good paper. Now, to your bioinformatics, let's suppose you found a drug molecules or potential chemical molecules which can inhibit the protein. So initially you publish a paper for that bioinformatic analysis. Now you can do some in vitro or wet lab based assays, and this may lend you a good paper. And that's how you can link the things. And now you can look into the structure of the protein, which is the protein which is coded by gene X. So when I take structure, when I say structure can be seen at secondary level, tertiary level, even at quaternary level. So you can use circular dichorism, you can use fluorescence spectroscopy, circular dichorism for secondary structure prediction, fluorescence spectroscopy for your tertiary structure prediction, and then you can do X-ray crystallography to know the quaternary structure or tertiary structure, or you can do NMR, nuclear magnetic resonance, to know the structure of your protein or your enzyme. Then you can do this cryo-electron microscopy. So there are you can do AFM is used to know the structure of other biomolecules, such as there are a lot of nowadays polymers and other nanoparticles are made. Even the structure of DNA you can see with the help of AFM, some DNA secondary structure, such as quadruplex and all. So there are a lot of techniques through which you can look into the structure. There is a lot of technique you can identify PTMs. Whenever I was talking about interacting partners, so like in interacting partners, you can do a simple pull down assay to look at what are the interacting partners of my protein. Then you can do mass spectroscopy, you can do mass LCMS. Then then other part is genomics. So if gene X is expressed, then what are the transcription factors which regulate gene X? So you can do the promoter studies of gene X. So you can clone the promoter in luciferase vector, and you can look what is the strength of my promoter, what are the different transcription factors which regulate my promoter. You can do chip PCR for this. And nowadays enhancers are getting a lot of attention, because one enhancer can control a number of genes. The theories are building up. A new field is getting popular nowadays, is nuclear condensates. So you can look into that if your protein is IDPs, in terms of its intrinsically disordered protein, you can look for potential that it do form some kind of nuclear condensates inside cell. You can go for DNA sequencing in terms that if you want to look that if gene X is related to any kind of disease, so you can look into that what are the mutations which are present in patients. So you just get a blood sample, you just isolate the DNA and look for and do the DNA sequencing, and develop some methods such as RFFP based, or any method that you can develop to look any SNP or mutations which are there in patients. So that's how you can do a research which is linked to even patient also, because try to collaborate with some hospital. If you're working with any disease and those kind of patients are there in India, you can collaborate with hospitals, you can get real-time samples, and you can do some DNA sequencing and RNA sequencing and other analysis to know that what are the changes actually happens or did happen at DNA or RNA level or at protein level in real-time patient sample. And then you can find that whether this gene has similarity with other genes. So this can help you to find out the function of that gene or with other non-coding regions. So this is one of the approach, or the 360-degree approach that I was talking about. So I guess I am I am done with this part of what to think before joining PhD. So Partha, this part is done.
Yes, sir. So shall I
Yeah.
forward
Yeah.
the questions?
Yeah.
Okay. There are a few questions has been asked by our participants. The first question is Nivedita is asking that how does a postgraduate student approach a scientist with their interest of to respond them?
Okay. So when you are in postgraduate, so a scientist always like that when you write things that I'm not making a biased thought, but sometimes a student is very enthusiastic to know about that technique, a particular technique. So when you are in postgraduate, you must tell them that I have learned this technique during my classroom and during my class, and I got curious and I learned more about this through YouTube and other learning channels. And now I'm more willing to get some hands-on and to at least look at instruments to correlate things more in better way. So it is very important that the scientists allow you to come to the lab, even in terms of to just visit the facility. So and then you can talk more because at first stage you must get a chance to visit that facility. So you must relate the thing that why you are interested in their lab, because you heard this technology, you learned more on this, then you have some because they still are growing up. So I believe that if you get a good lab in India, then it's always good to learn because you will get a lot of opportunity. You get more comfortable with them.
Okay, sir. Sir, Amita is asking that what about taking post graduation in biotech abroad?
Yeah, you can look for DAD fellowship, DAAD fellowship for Germany. There are a lot of other fellowships which nowadays in for Australia and Israel, and for in Canada. So if you are looking for MSR, or if you are looking for MS or MS in abroad, yeah, you can go outside, like in US. If you get a good department or institute in US, you can always go there because a lot of what do you say, like if you proceed in biomedical science or biomedical engineering, uh, or any course which deals with technique relevant to academy industry, you can get a good placement. So yeah, but try to get a fellowship. Try to know that what are the fellowships, what are the even yeah even it's very important now. Even you get admission in MSc in India, so there are a lot, or in PhD in India, there are a lot of exchange programs which occurs between institute and US or with the foreign nation. In IIT, when I was in IIT, there are a lot of exchange programs with Australia, with Japan, with UK. So you can fill Newton Bhabha fellowship, which is open for everybody. So and even you get a PhD and you want to in India and you want to do some part of your research in Germany, so there an exchange student exchange programs are there. You can apply in them and you can short-term visit. There are a lot of fellowships there.
Okay, sir. Sir, Aravinda Ghosh is asking that how we can determine the PTM using the instruments.
So post translational modifications, there are methods such as nano LC, mass spec nano LC. So nano LC, they can help you to identify ubiquit annihilation or somylation. Even there are other techniques such as Western blotting also. You can find some PTMs that whether, because there are some antibodies which can detect a phosphate labeled protein or non-phosphate protein. So, but nowadays, mass spec nano LC is gaining a lot of attention to identify these PTMs.
Okay, sir. Jill Charlie is asking that is life sciences students having any opportunity in defense?
Yeah. They can. Uh, they can go to DRDO. So because DRDO is Defense Research Institute, and there are other avenues which opens later in forensic science laboratories, not directly related to defense, but FSL or NIA, they hire any students or like who had done a good PhD, they can apply for this post senior scientific officer, which are there. Even Homi Baba Atomic Institute, they have this scientific officer post where you can apply and you can get in touch with different related activities.
Okay, sir. What bioinformatics tool can we use for plant pathology research?
So I'm not from plant background, but anything like you can first look for that. NCBI is the tool, and nowadays NCBI is made themselves so friendly that they show that what are the transcripts and what is the gene expression level, what are the mutations which are related to this gene in this plant pathology, which is, let's suppose any kind of pathology is there. So because I'm not yeah. So if you know the protein is responsible for causing some kind of pathology in protein, so you can look any intervention method to stop that protein so that you can stop the pathology. And bioinformatics becomes very important because we know that we can engineer seeds and all, so that we can and in this way we can use various bioinformatic tool to find out any siRNA based or any aptamer based technology to, or CRISPR Cas based technology to stop the pathology.
Yes. Slovan Raj is asking that how important is learning CRISPR as a new technique.
Yeah. Now the CRISPR is getting a lot of attention nowadays, and we know that the CRISPR can help you to silence a particular any gene that you are interested out of thousand gene. So let's suppose that person is interested to know the function of gene one. So through CRISPR you can silence even the promoter. Through CRISPR you can silence that gene, or you can even mutate things to know that what are the effects of that silencing in so if you silence gene 1, then what are the repercussions, or what are the ripple effects. So further he can do RNA sequencing to know that what are the global changes occurred in cell RNA level when I silence this gene. So CRISPR is very important. siRNA is also there to silence gene, but CRISPR technology learning this will help you to get a good PhD or a postdoc,
Okay, sir.
because it do not have a background effect that other techniques have.
Yes. Suganya Sundar is asking that how important is animal study in PhD?
Very, it is a very good question. That if you see that a lot of postdoc positions and a lot of faculty positions, sometimes they get advertised that having animal model experience becomes very is a desirable thing that they are asking. So not only animal model such as mouse, or if you are lucky then you can use chimpanzee and all. So like you can you can learn Drosophila, you can learn zebrafish, you can learn C. elegans. So these are these are the models which are always exciting to learn, so that they can get you a good postdoc or a good even in many institute in IIT, that sometimes if you look for those faculties which are having experience in Drosophila or C. elegans, so that we can approach them in terms that we can do some in vivo essays. So it's very important to learn any model is very important.
Okay,
So this is the thing which is going to add in your CV.
Okay, sir. So the last question of this session is, participants are asking that how can they connect with you for their future queries?
So yeah, I'm there available on Facebook. So I'm there on Facebook. You can mail me. So my email ID you can take. Like I can give you Partha. So but my email ID is Prashant 1400 @gmail.com. And I'm on Facebook also. You can find me Prashant Pradhan on Facebook, and you can just connect to me.
Okay, sir. Let me give the email ID to all the participants. Yes, now we can proceed with the second session, sir.
Yeah, but I guess I will love to take a 5 minute break.
Sure, sure, sure. Okay, sir.
Partha, I will try to. Should we start?
Ah, yes, you can start, sir.
Okay. I'm sorry to all that in between my voice is breaking and I was not audible. I'm really sorry. So, students who can look into this, they can see that this red trace is going by and passing through a glass and getting out from to air side. So what you can see is that light is getting what reflected or refracted. It is refracted. So, but when you see that, when I change the angle from red, so I'm changing the angle in a way that I have achieved the angle, so blue line, in a way that the light is not get refracted, it is get reflected back. So this is called a TIR, total internal reflection. So I guess you are aware of this TIR. And now, why I am talking about TIR, it becomes because I am talking about SPR. So, in SPR, what we do is it's just a illustrative example. I will talk more on this. It's just what you can see is that there is a glass plate and there is one prism. There is one prism over it, and in yellow you can see a gold plate. Okay, so there is a gold plate, there is a glass plate, and then there is a prism. And on the gold plate you can see some Y shape this ligand which are immobilized on this, which are attached to gold surface, so Y shape such as antibody. And then you can see this blue dots which are analytes which are flowing over this ligand. And so the other segment is you can see a light source, a red light is coming. So instead of getting refracted, it is getting reflected back. So if you see there are two, so in one case, when you see that, so this incident light got a minima, so intensity minima is here. So why this minima you get? So why this minima you get in SPR? And that this is the whole thing. This is a basic principle which is there. So surface plasmon resonance. So first, what kind of surface I am talking about? So surface the surface that I am talking about is the gold surface. Is the gold surface. So why we are taking this kind of gold surface? So we can take other surfaces such as indium, we can take copper, we can take nitrogen or this sodium, but there are limitations with those surfaces. So the surface that we choose for SPR is those surfaces which have free electrons, or the electrons which are available for conduction. Okay, so, and the plasmon is the particle name of the free electrons which are oscillating, and that the resonance the waves of free electrons, and the resonance occurs when the resonance occurs is, listen the line carefully. When the light is made up of what? Light has photons has energy. So at a certain angle, when I have achieved the TIR, and near to that angle, when the energy of photons, so because when this lights comes, it get reflected back, and you were getting an intensity minima. So that means some energy was lost during when the light was reflected back. So what is this energy intensity loss is that the photons were the energy of photons was coupled with the free electrons of gold, and this coupling lead to the resonance of those free electrons, and that lead to change of this reflected light with different minima. So you can see that there are two waves, so first wave and second wave. So first wave is a wave when there was so I will explain you everything, but so let's suppose I added something, I added these blue balls. So first wave is when there is no blue ball, so this is the first wave. But when I added these blue balls, the second wave is this. So when I added this blue ball, this lead to change in the angle. There is an angular shift of this intensity minima. So I will explain you everything. I am making you comfortable with this technique. So to summarize this, in SPR we have a glass prism. The glass prism, then we have a glass plate over which we are coating a gold plate. We are taking gold plate because the surface has free electrons, conducting electrons, and on gold surface we are immobilizing suppose antibody, and then a blue ball is your antigen. You want to see that your antigen interacts with your antibody. So let's suppose blue ball is your antigen and your this Y shape is your antibody. So when the light source passes without this antigen, it will have some angular, it will have some intensity minima. And then if this blue ball or antigen interacts with this Y shape antibody, you just see an angular shift. So this angular shift, you measure this angular shift. This change in angular shift is measured in terms of response unit. Let's now come to the graph. So change in this angular shift is recorded in terms of response unit. So this is baseline. Now you can see there is one term called association. Are you able to see this association? So what is this association? Initially, when there was no blue ball, blue ball, you will get the baseline. But when this blue ball starts interacting with this Y-shaped antibody, what you will see that there is an increase in the graph. So that the blue is interacting with Y-shaped antibody. So what you are getting an association. Okay. So this association, why there is an increase in this? Because there is increase in response unit, and this response unit is proportional to the amount of angular shift. So amount of angular shift in this before and after the binding of the blue ball to the Y-shaped structure gives you Ru, and this Ru will be that much, and it is proportional to the angular shift. So once the association is done, so let's suppose there are 4 Y-shaped antibody you can see in this. So maximum 8 or 4 to 8 antigen can bind to it. So once the maxima is reached, it will reach to equilibrium. So what is equilibrium? Equilibrium is a place where equal number of particles are binding or blue balls are binding and equal numbers are dissociating. So equilibrium is something which has graph forward jitna forward utna reverse. So equilibrium is something which is k on and k off is equal, and then there is a phase called dissociation, that I am removing every ball, and then what you will see that there is a dip in the resonance unit because the angular change is there. Now we will regenerate the surface. What is regeneration means? That any blue ball which was not dissociated at the time of dissociation, we will add some buffer so that this complete dissociation occurs and no blue ball is still binding there. So this is called regeneration, and again you will get the baseline. So I guess let me summarize this again, that what I am trying to tell you, that first thing, the surface plasmon resonance is based on change in refractive index. It is based on change in refractive index. Okay, so the light which was coming initially before any blue ball binds to this Y, so you were getting one angle, and when this blue ball binds to this Y-shaped structure antibody, you were getting an angular shift. So there were 2 graphs here. So the amount of shift depends upon the amount of antigen or blue ball binding to your Y-shaped antibody. So because when anything binds here, it will lead to change in refractive index. A change in refractive index will lead to change in SPR angle, which is angular shift. So higher the mass, higher the change in refractive index, higher the change in angular shift, higher you get a resonance unit. So I repeat it again. So when there was no ball, the refractive index was same, so you get a baseline. So when this blue ball binds to this Y-shaped structure, a refractive index changes. Change in refractive index will depend upon what number of mass or the number of molecules which are coming here and binding to this Y-shaped structure. So this change in refractive index is proportional to change in angular shift, and this change in angular shift is proportional to the increase in RU, and this RU is something that we note. So I guess I was able to make you clear. I will make it more clear in next slide. So what happens that when this light, so let's suppose, so let this yellow is a gold plate. So when this light passes is through this glass prism, it gets reflected back. So what I told you that during this process, what happens that the photons are carrying energy. So some of the energy get coupled with the free electrons of gold surface, and this coupling leads to resonance of these plasmons, and they send a wave called evanescent waves. So I hope this wave is word is visible. Evanescent wave. Evanescent waves are the waves which are the decaying waves. So they travel at the both side. They travel so if the waves are created here, so they will travel in this direction as well as in this direction, both in Y positive and Y negative. Now these evanescent waves are very sensitive to change in refractive index. So that was I was telling you that because the evanescent waves are traveling here and they are not visible, but and they are sensitive to refractive index, change in refractive index. So when this blue ball binds to this Y-shaped structure, a refractive index will change, and this will lead to the change in this angular resonance angle, and that's what we get a shift in angular angle shift, and that's what we record. So A is a wave before ball was binding, and B is a change when ball binds. So a difference between A and B is the change in angle. So now you can read it. The semantic experiment setup: surface plasmon resonance, a sensor chip with a gold coating. So this is your prism, and this is yellow part is your gold coating, and this is your light source. So a polarized light. So very important, the light source is monochromatic and it is polarized, plane polarized. So reflected light intensity is measured in detector. A certain angle of incidence, which is this angle, excitation of surface plasmon occurs, resulting in dip of intensity. So this dip was there because some energy was transferred from photons to the free electrons of the surface. That's why there was a dip in the intensity of the reflected light. Now the intensity, a change in the refractive index at the surface of the gold film at this surface. So I'm talking about this surface or this surface. So change at this surface will cause an angle shift from A to B. So I guess I'm able to make you clear that a change from A to B, why it happens, because there is change in refractive index, and that's why it happens. And refractive index is linear to amount of substance which is binding. So this is very important. So this diagram will make this clear. So this is light passing on, and this is metal film, and this is a detector, and this which is detecting a reflected light. So in first case, where this is the dark portion, this is the dark portion, original dark position. But when this molecules binds to, so this is ligand, and this is the biomolecule which binds. So before binding of the biomolecule, this was the dark line. But when this biomolecule bound to this ligand, you can see that this dark line shifted towards your right. So this is called angular shift. So at certain incident angle or resonance angle, the plasmons are set to resonate with light. Okay. But if the refractive index changes, then the resonance angle will change. So the resulting in the absorption of light at that angle. This creates a dark line in the reflected beam shown in figure 2. So this is how a simplistic diagram which looks. So this is the light source which is getting reflected back, because here we have what? A gold surface. Gold surface has free electrons which can take up the light energy, and this light energy can coupled up with this free electrons of gold surface, and then they send evanescent waves at both the side, and these evanescent waves are sensitive to change in refractive index. So amount of analyte binds to ligand will be proportional to change in the angular shift. So this is a chip. So chip has a glass plate. So this is a chip that we fit into SPR machine. So chip looks like this. This is a gold plate, a small gold plate, square like structure. So here we have a glass, then we have a gold layer, and over we have a dextran matrix. We coat the gold with dextran matrix is just because the dextran provides a amphipathic environment and it can help in binding of biological or biomolecules. It's just for binding, for absorption of those biomolecules that we want to test. So what all type of experiment I can do in SPR? So let's suppose my protein, so let's suppose this is my protein, which has histidine tag. So sorry. So this is my ligand, or which is protein. Let's suppose protein X. So let's make an experiment. I want to check that it will be easy okay, so this will be, so I want to let's make a imaginary example that I want to see that whether my protein X interact with protein Y. Okay, so I want to check interaction of protein X with protein Y. So what I am doing, that I am immobilizing a ligand which is protein X on gold surface, and then I will flow the protein Y over this. So if protein Y interacts with ligand, it will lead to change in angular shift. So first for this, I am immobilizing my ligand which is having a histidine tag. We know that we purify the protein with histidine tag, and with the help of nickel NTA chip, we immobilized, we were able to immobilize our ligand on this. Now, what will happen? So let's suppose in this diagram, my ligand, which is protein X, is immobilized on this gold surface. Now, when this protein Y, which is red color, binds to this surface, what will happen? That association will occur, and association will occur to some maximum where all the positions which were there, they got occupied and equilibrium is reached. Now I gave a dissociation, and then I again regenerate the surface. So these are the basic steps that I will repeat again and again in SPR. So through association and through dissociation, you can find very important term which is called kinetics. So how fast? So when I say kinetics, I take time into consideration. How fast my protein Y interacts with protein X, and how fast or slow it dissociates. So let's suppose I discovered through bioinformatics, I discovered a number of drug against protein X. So what I will do, first I will immobilize the protein X on gold surface. Then one by one, I will flow my drug 1, drug 2, drug 3 over the surface, and I will check the kinetics, that which drug was able to bind with high rate of kinetics, because in terms when we are discovering drug, it is very important for a drug to have a good dynamicity, because if we cannot afford a drug which goes and binds to the molecule forever. We want a reversible reaction, and we want a fast dynamics in this. In the biological system, dynamics is very important. Just an imaginary example. So we can read the kinetics, like what is the K on means that what is the association rate. Rate means that how many molecules are binding to my ligand in per unit time, and how many molecules are dissociating per unit time. So this is the kinetics. And then an important thing, which is equilibrium. This equilibrium where we have equal K on and K off is help us to find what? Binding affinity. So three main parameters you are getting. First, association rate. Second, dissociation rate. And third is the affinity. Actually, how much is the affinity in molar? It is in molar range, it is in micromolar range, or it is in nanomolar range. So biological molecules which have high affinity, they fall in nanomolar range, and the drugs, they fall from nanomolar to micromolar range if they have a good affinity, and sometimes if they can go to millimolar, but they are not good. So nanomolar binding type of affinity you can quantitate through SPR. So what we do actually, is you can see that what association is that when A plus B gives AB, that means your ligand and analyte binds it gives AB. So the reaction rate of AB you can find, and dissociation is when AB, which is formed, it is dissociated. AB dissociate to A plus B. And the second parameter that I told you was equilibrium association constant or equilibrium dissociation constant. It means the affinity constant is that Ka divided by Kd. So number of in this it's just a reverse of this. So Ka divided by Kd gives you K capital A. Now many students make some mistake. This is K capital A, and this association rate was K small a. Here I talk about equilibrium association constant, so I am talking about this region where Ka and Kd I am talking about when they were in equal range. The rates were same. So this is the thing, Ka divided by Kd, or K capital D, or the affinity constant or the dissociation equilibrium constant is K small d divided by K small a. So remember these small or capital letters. They makes lot of difference because one is binding affinity gives you in mole and other is in time, second or per second. So kinetics and affinity you can find. So the basic principle is that the SPR signal originates from the changes in the refractive index at the surface of the gold sensor chip. The increase in the mass associated with the binding event causes a proportional increase in the refractive index, which is observed as change in response that I told you. These changes are measured as change in resonance angle or of refractive light when the analyte flowing in a microfluidic channel binds to the immobilized ligand and increases the density on the sensor chip. So importantly, for protein-protein interaction, the change in refractive index on the surface is linearly related to number of molecules binding. So that's why we can calculate how many number of molecules got binded. So stoichiometry we can also calculate from SPR as stoichiometry. So first is kinetics, second is affinity, and third is stoichiometry. So this is the real-time SPR graph that, and it is from my work that. So let's suppose I take an example that I immobilize 50. So in a room there are 50 chairs. Okay. So this black trace. So this black trace. Let's suppose this first black trace. In first concentration, I allowed only 5 students to come in class. So what will happen, because seats were abundant, so ligand was 50 and analyte which came, they were only 5. So they will bind and they will reach the equilibrium and then they will dissociate. In second concentration, what I will do, I allow 10 students, so they will bind, they will sit in the on chairs and then they can leave. Similarly, I increase the number of students till the time when seats were 50 and I allowed 50 students. So what will happen, that the response will reach to the maximum. So association as the number of molecules which are bound, which they will increase in the resonance unit to the maximum and they will reach to a saturation and then they will dissociate. So if I allow more students, so what will happen, that only 50 can bind, not, and other 10 will not bind. So what you can see that you can see the overlapping. So this magenta and blue, they are overlapping. Even I am increasing the concentration from 1000 nanomole to 1800 nanomole, they are not increasing. This means that the seats were full, the seats are full, the binding is full. And when you can plot this, so this is the maxima, black is the maxima here of this, red is the maxima of here, green is the maxima of here, and I get a plot, and through this plot I can find the Kd, K capital D, which is affinity, and I got 625 nanomole because it is 10 to the power minus 9 moles, and 203 nanomole. So what is the difference is that this molecule, so this binding, so in this case I immobilize the prion protein, I immobilize the prion protein on a gold plate, and I use 2 DNA molecule, A and C. So you can see that A is binding with 625 nanomole and C is binding with 203 nanomoles. C has more affinity to my prion protein, and you can see that there is a difference in association and dissociation also. So in this way you can calculate the kinetics, you can calculate the KD, and this binding was at 1:1 ratio. So 1:1 was the stoichiometry that I found. So this is the real time example that I used to make it to make you more connected to the real time experiment. So I'm done with this. So questions are welcome. Hello. Hello.
Yes, sir. Sir,
Yeah.
you can
So
proceed.
hello.
It's audible.
Yeah,
Wait,
Partha,
wait.
I'm done with the session. So any question related to SPR, you can
Yes, yes,
take.
yes, yes. Wait, sir. Sir, I'm proceeding with the questions. Sir,
Mhm.
yes. How SPR is different from ESR?
Mm, I have not done ESR, so sorry, I'm not able to tell you about this.
Okay, sir. And another question is that, sir, you are saying angular shift. How do we measure the angular shift? Angular shift can occur due to movement of surface, or we measure the energy shift of reflected electromagnetic radiations.
So angular shift, the change in angular shift occurred because there was change at this level. So change and this level change led to change in refractive index, and because of change in refractive index, we got angular shift.
Why is the regeneration gap graph first lower than the baseline?
Okay, so regeneration when it just a sudden dip, just everything when get removed, it just a arbitrary sudden dip, nothing much.
Okay. Can this SPR be applied to detect plant viruses?
Exactly, it can be.
Okay. What?
Even you can immobilize a whole cell. So this is something that so you can immobilize a whole cell on the surface, and then you can look the interaction of other thing that whatever you want to do.
Okay. What type of enzymes are used to mobilize ligands of protein?
So immobilization can be on basis on amine. So our proteins have amine group like lysine, arginine. So these amine can be coupled through amine coupling method. So there is this was amine coupling method on dextran. Then there was the other method called nickel. So we can also do it through streptavidin. Let's suppose I have my protein or my DNA bound to streptavidin, and then oh, so sorry. My chip has a streptavidin, and my DNA and my protein has a biotin label. So biotin and streptavidin can bind, and this way it can immobilize. Similarly, in this case, my protein is having histidine tag, and it is binding to nickel NTA. So there are a lot of other methods which help the protein to immobilize. It can be amine coupling, it can be nickel NTA based, it can be streptavidin biotin based.
Okay. Sir, what is the difference between capital A and a small a? KA and a
So
small
K capital A gives the affinity constant. Okay, the amount of binding which is there. So what is the amount of binding is there, and K small a gave you the rate of how many molecules were binding at a particular time. So Ka gives you association rate, and Ka gives you affinity constant. What is the affinity of that binding?
A? Is which define in which field we can use SPR?
In any field, like if you want to check that what is the what is protein binding affinity and hello.
Yes, yes, yes.
Yeah, my screen is gone, but I guess I'm audible.
Yes, yes, it's
Yeah. So in any so if you want to check that what is the affinity of my enzyme with any molecule, what is the and what is this how many and what is the affinity of protein X with protein Y, what is let me get back to screen first.